Okay, good morning. My name is Alexis Battle, and I'm a professor of biomedical engineering at Johns Hopkins University. And today I'm going to talk to you about machine learning and genomics. So we'll go through a few parts. I'll talk about what is machine learning, and then I'll give a bit of a background of different approaches that are used in machine learning, especially in genomics. I'll talk about some common pitfalls and considerations that come up, particularly in the applications of machine learning and genomics. And I'll give an example of a method that we developed in my lab. So first of all, just to start with the very basics, what is machine learning? So machine learning, as I define it today, is the computational identification of patterns from data, which can then enable predictions or decisions from an automatic system. In slightly more technical terms, I think of this as automatically training the parameters of some system, such as a decision rule, a prediction function, or a model based on data. And the real key to both of these definitions is that it's automatic, so it's a computational and automatic process, and that it's from data. So data is going to be a real key to everything we talk about today. So the definition that I give is in contrast to two things. So I said it was about computation and about data, and that's in contrast to human specification of a method or a function. So human specification of heuristics or human specification of the parameters of a prediction function would be in contrast to what I defined as machine learning. And second, for a long time, AI methods also used hard-coded rules, so not based on data, not based on machine learning, but simply encoding an automatic system, but based on hard-coded rules instead of parameters or rules that are derived from patterns and data. So one aspect that gets confusing in the field now is what exactly counts as machine learning. So there's methods that are from statistics or data mining that some people aren't sure if we should really refer to them as machine learning. So for instance, is a simple univariate linear regression machine learning? Is multivariate linear regression machine learning? Is every automated analysis of data, going to fall under this lump of machine learning? So I'm going to be really inclusive in my usage of the term. So, no strict boundaries here. I'm not going to try to draw a sharp line between statistics and machine learning. I'm going to be very inclusive and really refer to every automated identification of patterns from data as a form of machine learning. Without any argument today, I'll just be very inclusive. So why are we using machine learning and genomics so much lately? So I'm going to argue that the time is really perfect for the application of machine learning and genomics for a few reasons. So first of all is the availability of data. So in the past 10 to 20 years, we've had an explosion in the types, the accuracy, and the breadth of data that's being collected at scale. So from efforts like UK Biobank, all of us, we're now getting millions of individuals for whom we have genetic and genomic data. We're collecting new types of genomic data from epigenetics to single cell expression and other functional data types to more accurate genomic data from DNA, including long reads. So that we've really collected this huge wealth of data, both in diversity, accuracy, resolution, and number of samples available. So we have the data. And second, the machine learning world has provided us with methods that are appropriate for these large-scale complex data such as genomics. So we now have methods that are appropriate for very, very high dimensional prediction problems with not only many samples, but millions of features. So we have methods that can address that. We have methods that are capable of analyzing high dimensional time series, high dimensional spatial data, and others. And of course, there's been great developments in recent years in applications of neural networks to large high dimensional data. So we have the data and methods development. In addition, there are many applications with pressing needs where machine learning and genomics can make a difference. So we're interested in everything from genetic risk prediction in a clinical setting to methods for integrating complex multi-omic data, so multiple different types of data together, to problems of cell type identification, patient subtyping, gene network inference, and analysis of genetic ancestry. So we have data, methods, and we have needs. And I would point out that these needs that I've identified are complex enough that we know that methods that are not based on data are unlikely to work. We cannot hand specify methods for genetic risk prediction or for multi-omic data integration. We as humans cannot simply sit down and look at the output of these large-scale genomic assays and identify rules that would predict the quantities that we care about. So the time is really perfect for the application of machine learning and genomics now. So I'm going to go into a little bit of background in machine learning methods now and some basic terminology. So first, we're going to cover different categories of machine learning. The first that I'll talk about is supervised machine learning. So supervised machine learning is defined by a goal of automatically learning some function to predict an outcome of interest from a set of measured features. So we're really trying to learn some function f, the parameters of such a function that can predict what we care about from measurements that we're able to take. So for example, I've listed a couple of examples outside of genomics. So the first would be face recognition. So we can take photos of people now, a picture of a person, and try to predict the identity of that person. Who is the picture of? So here the features would be just the grayscale or color pictures in a photo of their face. So just numerical values representing the image itself. And the outcome that we're trying to predict is the name or identity of the person in the photo. So computer vision has become a very successful application of machine learning. We see this all over the internet and applications now. Another simple example, just to give you an example of the diversity, is we may be interested in predicting the price of a house. So here the outcome would be the numerical price of a home. And we would be interested in making this prediction based on where the house is, the number of rooms it has, how old it is, the size of the lot, and other attributes. So here our measured features are all the facts that we know about this home, and we'd like to be able to, from that, predict the price. So those are a couple of examples outside of machine learning to build some intuition, outside of genomics to build intuition. But here are some examples of supervised machine learning tasks in genomics. So a classic example would be disease risk prediction. So in this case, we'd like to predict whether an individual will develop a particular disease, so that's our outcome, from their genetic information. And here I've given an example of using single nucleotide polymorphisms, or SNPs, at 5 million different positions in the genome as our features. So here we would be given somebody's genetic profile, these 5 million SNPs, and we'd like a function that would predict their level of disease risk from that. Another problem that many people are working on now is epigenetic state prediction. So here we are interested in looking at a particular region of the genome and predicting its chromatin state. Will it be open or closed chromatin, for example, and a particular cell type or types? And we'd like to make this prediction based on the genetic sequence in that genomic region. So here the features would actually be the sequence of A's, C's, T's, and G's that we have in that region of the genome. And the outcome that we'd like to predict is, will chromatin be open or closed in a set of specific cell types at that region? Another example that's been a classic problem in genomics is predicting disease subtypes. So for example, if we have gene expression profiles from a tumor, so here our feature would be gene expression level at every gene, we'd like to predict a cancer subtype from that information. So those are some examples in genomics. So I'd like to just visualize what the data setup looks like for a supervised machine learning task. So our training data for a supervised machine learning approach, we need to give it numerical data. So we have to encode the information that we derive from our electronic health records or from our RNA sequencing run or whatever it is that we have available as our features and our outcome. We need to encode them as random variables and numeric values for all of our features and for our outcome. So this is kind of what a data matrix would look like for training a supervised machine learning task. In this case, I've given the example of predicting disease status from genotype. So in our feature matrix X, what we have is a matrix that is a number of individuals in each row, so one individual per row for n individuals. And in each column, we have the genotype at a particular SNP in the genome. So it's number of individuals in rows and number of SNPs measured in columns. And notice that we have to make some decisions. So we have to decide how to represent single nucleotide polymorphisms here. We're not simply throwing in the actual A's, C's, T's, and G's, but here we've made a particular decision to use the dosage of a minor allele as representation of these features for our SNPs. So if an individual is homozygous for the major allele, we encode it as a 0. If they're heterozygous, we encode it as a 1. They have one minor allele. And if they're homozygous for the minor allele, we encode it as a 2. But that is a choice. So this represents an assumption that we have sort of a linear effect of dosage in our system. We may choose to encode it in a different way if we're interested in capturing dominance and recessive relationships, for example, or something different. So we've made a choice of how to encode our feature matrix X, and we've laid that out. And then we have our disease vector Y. So here we have for each individual, so our 1 through n individuals, again, we choose how to encode their disease status. In this case, we've chosen a binary encoding, 0 for they don't have disease, 1 for they do. Of course, in many cases, we may have a more quantitative measure of disease, such as somebody's BMI may be encoded as a real value. And other cases, we may choose to use other encodings. In this case, we've used a binary encoding. So we set up our data matrix for our training data where we have observed both the features, our SNPs, and the outcome, in this case, our disease. And from this training data, we hope to learn some function, f of x, so a function applied to our SNP information that would allow us to predict our outcome y, in this case, our disease status. So this is our training data where we have available both genotype and disease status. But of course, for a test case or a clinical application or future use of this prediction function, we would only be given the information about genotype or only the features X, and we would actually be predicting disease status from scratch in that case. But again, when we train, we have both features and outcome, X and Y, and when we predict, we only have available X or the features. So one thing I want to point out, particularly in genomics, is that there may be different goals in mind when you decide to build a supervised machine learning system. So you may be motivated, for example, by accuracy. You really care about making good predictions in practice. And this might be the case if you're thinking about a clinical application or a translational application where what you really care about is that your predictor is highly accurate for practical purposes. But you don't necessarily need your predictor to be interpretable. You don't need to be able to look at that function f or the parameters of f and understand how it works. You'd be just as happy with a complete black box as long as it's highly accurate. So in this case, you would care more that it's correct and care less about what it means. But sometimes in genomics, we do build predictors when we care more about interpretation than accuracy. So we want to be able to inspect our learned function f to determine what features influence the outcome. So we may want to know, for example, which genes are most relevant to the disease that we're predicting. And in this case, we care that the model is meaningful and interpretable, and accuracy may be actually secondary. In some cases, you have both goals. You care both about accuracy and interpretation, but in some cases, you care much more one about the other. And this may imply different methods would be appropriate for your problem. And we'll come back to that in a moment. So when you're choosing a supervised machine learning method, that's basically deciding what type of function f will I be interested in learning from my data. So there's some considerations. For instance, how expressive does that function f need to be? What types of relationships do I want to capture? Am I only interested in linear and additive effects? Do I care about complex patterns and interactions between my features? So for example, a linear function f would only learn additive effects where SNPs act independently to create risk for phenotype. Whereas we may actually care about epistasis or complex interactions between the SNPs that affect the phenotype, and therefore a linear model would not be appropriate in that case. So ability to express complex patterns is one aspect that we need to think about when we choose a method. Another, of course, is interpretation. As I mentioned on the last slide, are we going to be able to take our function f and use it to extract biology from our learned predictor? How important is accuracy versus interpretation? And one aspect I want to point out is, do we need a probabilistic interpretation? Do we need to be able to quantify our uncertainty in our predictions, for example? And finally, of course, is computational efficiency. So I've listed a few machine learning methods for supervised tasks that are popular specifically in genomics. So the first are simple regression methods such as linear and logistic regression. These are very efficient. They are interpretable in terms of what they are saying statistically about the relationship between the features and the outcome. Another is support vector machines. These are efficient and give quite good performance in practice in many cases. They are flexible. A simple support vector machine will be able to capture linear and additive relationships, but more complex kernels and support vector machines can allow us to encode interactions and other complex patterns in the data as well. Another popular approach is random forests. These are very expressive. They can capture complex relationships. They have quite good performance on many tasks in genomics. They are somewhat more challenging to interpret simply out of the box, but methods have been developed for inspecting the results of random forests to tell us which features were most important in the predictive accuracy of the method. And finally, I list supervised neural networks. So neural network methods can be appropriate for supervised tasks. I'm going to come back to neural networks later in the talk and discuss them in more detail, but I will, for comparison here, point out that they're highly expressive. They can encode complex relationships between features and the outcome. They are more computationally demanding, so they're a bit less efficient. And in their basic form, again, these are more of a black box method that is less interpretable. But here, methods are being developed for inspecting neural network models to offer some interpretation of the features and the properties of the data that lead to the predictions that they make. So to give an example of a supervised machine learning task in genomics, here I'm specifying just what the data would look like and what we're trying to predict. So here the goal would be to predict whether a colon cancer patient will respond to a particular drug based on gene expression of a tumor. So the training data here would include RNA-seq for some number of patients, so N patients, and for all of those patients, a record of which of them responded to that particular drug. So we would actually need training data where we had tested this drug in a number of patients and knew which patients responded and which didn't. So our features X, so again, we need to encode our data numerically. So our features X would be real value expression levels of some number of genes, perhaps genome-wide gene expression for K genes measured by RNA-seq for all N patients. And in this case, a good outcome would be perhaps binary representation, so 0 or 1, representing whether or not that patient responded to that particular treatment, when we'd need that information for all N patients in our training data. And a candidate approach that we might be interested in trying is a support vector machine to separate responders from non-responders based on that RNA-seq information. And what I'm showing on the right here is a very simple graphic where we're assuming we have two features or two genes, and we're looking for a hyperplane that separates responders in blue from non-responders in magenta here. So a simplified example. So we're going to move on now to another category of machine learning approaches called unsupervised machine learning. So recall in supervised machine learning, we were trying to make a prediction of a particular outcome. So unsupervised machine learning is different. This refers to a set of statistical tools where we've measured data or features. Again, we have some number of features and some number of observations. But in this case, we don't have any labels or a particular outcome Y available for our training data. And the goal here is to discover interesting patterns in X without a teacher, without any supervision at all. So we have no response variables or labels. So why would we want to do this? I've given a few examples here. We may be interested, for example, in seeing if there's an informative way to visualize our very high dimensional data. In genomics, again, we often have thousands or even millions of features, which we cannot simply visualize all together in our population. But using unsupervised machine learning methods, we may be able to come up with a visualization that tells us something informative about our data. And a particular common task is clustering, or specifically, can we identify subgroups among our variables or among our observations, groups of features or groups of observations that go together in some meaningful way. So for both of these tasks, we don't need any label. We don't need an outcome that we're predicting or that we have available during training time. So I will say that unsupervised machine learning is often a more challenging task than supervised machine learning. I don't mean computationally, I mean scientifically. So it can be hard to assess the results from unsupervised machine learning. We don't have a labeled outcome, so it's hard to get sort of ground truth of whether an unsupervised method has done a good job or the best that we can do. It can be a more subjective task than supervised machine learning. So we may be looking at our visualization and simply saying, oh, yes, I like this one better. So those are some of the challenges involved in unsupervised machine learning. And I will say that it's often performed as part of an exploratory data analysis. So the first category of unsupervised machine learning that I'll discuss is called clustering. So here our goal is to identify discrete groups of similar entities in the data. So for example, we may be trying to cluster genes and identify genes that appear to work together according to some data, or we may be trying to cluster patients. So for example, to identify patients whose tumors behave in a similar way according to gene expression. And I've listed some popular methods that are used in genomics, including k-means, Gaussian mixture models, graph-based clustering methods, and a very common hierarchical clustering approach. Separate from clustering is an approach called dimensionality reduction. So in dimensionality reduction, it actually has a very related goal to clustering, but instead of identifying discrete groups or actual like clusters of individuals or clusters of features, we're simply trying to find a lower dimensional representation that captures the main patterns and main relationships in the data. So one way to think about it is instead of trying to find, discrete clusters that genes fall into is actually to find patterns of gene expression that explain the majority of genes' behavior, but to find a small number of those patterns that capture the behavior of most genes. So methods here that you may be familiar with include principal component analysis, but there are many more, such as many diverse forms of matrix factorization, such as non-negative matrix factorization, in addition to other nonlinear dimensionality reduction techniques such as UMAP and t-SNE. So these types of dimensionality reduction methods can enable interesting visualizations, interpretation of our data, and identification of representative trends, so trends that many examples tend to follow. An example scenario where we may be interested in using unsupervised machine learning in genomics is in single-cell genomic data interpretation. This has become very important. So for example, if we have measured single-cell gene expression from brain tissue, so we've measured perhaps P features, gene expression levels, and we are interested in discovering unknown cell types or unknown groups of cells. So in the brain, we know that there's very complex cell types. There's, not only neurons and glial cells, but there's many subtypes of neurons. We may not even know how many subtypes we're looking for. So we begin with a set of gene expression from what I'm calling mystery cells. So we just have a bunch of cells and their gene expression. And our goal here is, in an unsupervised way, to group those cells together into clusters. And in this case, we may ultimately get out clusters that are biologically meaningful, such as different neural and glial subclasses. Alternatively, even from that same data, we may be interested in discovering not simply groups of genes, as in this slide, but actually patterns of cell type and cell state. So for example, we may have a case where we have not only multiple different cell types, but actually a continuum of cell states, such as position in the cell cycle, or differentiation time, or other states that are not simply a discrete membership in a cell type class. And in that case, we can apply dimensionality reduction techniques such as UMAP or even just PCA to that same gene expression matrix that was shown in the previous slide. But in this case, instead of outputting clusters, we're outputting information about the prominent trends in our data, or in this case, a visualization using UMAP that shows, for example, every cell in our data, what cluster, how they seem to relate to each other, in this case, laying them out so that cells that appear to be more similar together are placed near each other in this two-dimensional visualization based on a high-dimensional original data in matrix input. We get a low-dimensional representation that is conducive to visualization out. Okay, the final methodological class that I definitely want to mention is deep learning and neural networks, which you've probably heard a lot about in the news and in the literature in recent years. So what is a neural network? We'll start with the very basics. So a neural network is a complex model that's based on the combination of many, many simple operations composed together into complex networks. So we start with a simple neuron model, so a single node in a neural network that takes its inputs, whichever nodes or inputs happen to connect to that one neuron, and performs a very simple function on those inputs to provide its output, which it then propagates onward to the neurons in the next layers. So deep specifically refers to the number of layers of these nonlinear operations that occur. So one neuron, we can look at a single neuron and what it does. So I'm here discussing the sort of origins of neural networks, and one neuron is a perceptron. So a perceptron takes a set of inputs. These are just like our features X that we talked about throughout the supervised and unsupervised learning sections. So a number of inputs, and performs a simple operation on them to produce an output for that one neuron. So generally, it's taking, first of all, a weighted linear combination of those inputs. So we have our inputs X times, weights W that are learned as part of that perceptron or part of the larger network. That's simply summed together, but the key is that we must add a nonlinear function in there. So to make a neural network interesting, we have to have nonlinearity. If we had this huge deep network where every neuron did nothing but apply a simple weighted linear function, then we have nothing more than a linear function as a whole. So the final output of a deep network that was purely linear would simply be a linear function of the original inputs, and we would not have gained the ability to represent these complex predictors or functions using our neural network. So each one of our perceptron nodes applies some nonlinearity to that weighted sum of its inputs to produce an output Y that's propagated on to the next neuron in the network. So for example, a nonlinear function that may be applied is a sigmoid function, but there are many others available as well. So from these individual, very simple single neurons that apply a simple weighted sum followed by some nonlinearity, we can compose highly complex neural network models. So I'm not going to go into the details of all of these, but we can have different functions performed at a given neuron. So they may apply a convolution operator, they may simply pool and take the maximum of a set of neurons in a layer before them. They may apply different nonlinearity functions. We may have fully connected layers where every output from one layer is connected to every neuron in the next layer, so that's fully connected. So there's many different choices that we can make at each layer in terms of the structure, meaning what neurons are connected to other neurons, and the operators that are applied. And from this, we can construct very, very complex networks from these very simple individual units. So what is a deep network then, right? So we've laid out what it's doing sort of mathematically, but what is it doing as a whole? And there's different ways of thinking about this. So one useful way of thinking about a deep neural network is as a universal function approximator. So a deep neural network can approximate a large class of complex continuous functions by learning weights over combinations of these simple operators. So an arbitrarily complex continuous prediction function that you could define over your high dimensional complex data, a neural network of this simple basic form has been shown to be a good approximator of this class of complex functions. Another way to think about it that comes from my training in probabilistic graphical models is that it's just a really, really big probabilistic graphical model with lots and lots of latent unobserved variables. So if you go back and look at what a network looks like, we begin with input from our data, and then we have this many, many, many layers of neurons that correspond to data that we haven't observed. Maybe they're capturing some pattern in the data that's a combination of original features from our input data, but it's being processed along the way in these latent nodes that don't correspond to a direct observation. So you can think of it as a big probabilistic graphical model with lots of latent or unobserved variables. And a final way to think about it, and really some of the inspiration for neural networks, is that it's a model mimicking or inspired by natural biological neural networks, and therefore hopefully capturing some of what we're able to do. So here are some pros and cons if you're thinking about, should I be using deep learning for a particular problem that I'm working on? Some of the pros are automatic construction of highly complex features, or basically automatic detection of complex relationships in your data. So it can go way beyond sort of a linear additive model and learn very complex features of your data that may be related to the outcome that you're trying to predict. And you don't need to know anything about those features ahead of time. So for example, in image recognition, I can simply give a deep network numerical representation of the pixel values in an image of faces, and neural networks will automatically construct internal neurons, these latent variables that correspond to things like edges in the picture and the presence of an eye or a nose. But I didn't have to tell it anything about that at all to begin with. I just gave it these raw pixel values. Whereas if I wanted like a linear model to use any features like that, I would have to construct edge detectors or nose detectors and input those as features. Neural networks can learn them automatically. And you can see similar things happening in the application to genomics, that genomic deep learning models can automatically learn things like transcription factor binding motifs and other complex attributes of the data automatically. So those are some of the big pros and why many people are using them. Some of the cons, or at least things to think about, is that neural network models are very data hungry. They can be prone to overfitting to the data because of the complexity that they can capture, and they can be challenging to interpret. And of course, with really large models, the computational cost can be higher. So why have neural networks gained popularity in recent years? So neural networks have been around for decades, but suddenly in the past, 10-ish years, they have achieved a level of success that's really never been seen before. So how has that happened? I would highlight, these are not the only reasons, but I'd highlight three things. One is improved computational power and use of GPUs. Another is new neural network architectures or designs. So approaches that you may have seen in the news, such as transformers, diffusion models, and graph neural networks, are enabling us to more effectively capture complex relationships in particular types of data, including sequence data. And the last that is definitely relevant for genomics is data availability. So neural networks are data hungry, and when we try to apply complex models to small numbers of samples, they weren't as successful. But as we've moved to a world where we have millions of examples available, we are now well powered to learn some of these complex models. So for example, in genomics, deep neural net approaches have been successful in problems such as predicting epigenetic state directly from sequence. So here, for example, we encode our input in a one-hot encoding approach. So for every position in the genome, we encode which base is present, an A, C, T, or G, using a matrix such as the one I'm showing here. And we can use a convolutional neural network that looks at these input sequences and identifies patterns such as motifs, through automatically, we don't have to tell it that, automatically identifying these in the neural network, and then using that information in these complex architectures to ultimately predict epigenetic state. But there's been really an explosion in deep learning applications in genomics, and here I'm just showing a few. So it's everything from direct from sequence prediction of epigenetic state and pathogenicity, to applications in single cell models, to building models of gene expression across cancer, and more. And I think that trend will continue in the coming years. So I do want to highlight what are some of the challenges and pitfalls that we face applying machine learning in genomic data. So the first one is still sample size. So in genomics, compared to many other machine learning applications, in genomics, we often have many, many more features available, such as SNPs in the genome, than we have samples, such as patients. So for example, when we were starting to work in GWAS, we had maybe hundreds or thousands of individuals whose genome we had measured, compared to, millions of SNPs or common variants that are present in those genomes. So building a machine learning model when you have way more features than samples can lead you to overfitting. So what is overfitting? I'm trying to show an example here on the bottom. So overfitting means that we are able to really, really accurately fit our training data using potentially a complex model, but it's overfitting, meaning that that's not actually a good thing and can lead to bad performance on unseen samples. So take the left-hand plot where I've drawn a set of training data in blue dots and a fit of a model in purple. So the left-hand model really, really accurately fits my training data. So I came up with a predictor that goes through, nearly perfectly every single point in my training data. But imagine if I had another point from the sample, a new unseen sample that I'd like to make a prediction on. What are the chances that it actually lies on that line? I would argue that the right-hand model that fits a simple line through these samples, given that we only have a handful of samples, is more likely to be accurate than this very complex function. So on the left is an example of overfitting, which can happen when we have many more features than samples, for example. So what do we do about overfitting in genomics? Well, first of all, you can select a simpler model with fewer free parameters. As I did in this example, a linear function may be a better fit than a high dimensional polynomial in this case. So fewer free parameters and a simpler model can help. If you're really limited in sample size, for instance, a neural network may not be your best bet. You can perform feature selection ahead of time. So from your millions of SNPs, you may cull them, performing feature selection to select a small subset to build your predictor from. You can use cross-validation to select a model that performs well. So you can hold out a subset of your training data and ask, how well does my model perform on data that it wasn't seen on? And for instance, that might show you that a linear model does much better than a higher order polynomial in this case, and you would select the simpler model using cross-validation. And there's other approaches such as shrinkage or regularization, and of course, ideally, just more training data. Another major pitfall in genomics is confounders. So unfortunately, genomic data, and especially functional genomic data, is affected by both biological and technical artifacts. So for example, if we're predicting disease status based on confounded gene expression data. So for example, perhaps we have collected all of our control samples in one batch and all of our case or disease samples in another batch. Well, then batch or sequencing batch is highly correlated with disease outcome, and our machine learning approach may simply identify batch, which is definitely not going to perform well when we try to apply it to new data for an individual collected outside of these batches. So these confounders create spurious correlations in our data and can lead to apparently, what look like really good predictors in our original data set, but that are simply due to confounders and won't perform well on new data. So I do want to highlight that there are so many factors that can create confounders in our data. It's not just batch, but how that sample was processed, what was the humidity in the room that day, sequencing artifacts, and then biological confounders that we may not want to capture, such as sex or genetic ancestry, environmental factors, or others that are not the attribute that we are currently interested in or trying to predict. All of these can create confounders in our approaches. So how do we handle that? So first of all, keeping track and good record of major technical and biological covariates in your study design is really important. But also we can apply machine learning methods to actually try to address this as well. And then of course, careful evaluation and ideally replication of your results in independent data sets is critical. But I do want to highlight that we apply unsupervised machine learning methods regularly to find confounders in our data. So we regularly apply PCA or related methods such as PEER to decompose expression data into components to look for factors that are highly correlated, not with our variable of interest, but with things like genetic ancestry or technical artifacts like batch. And the loadings that we get out of PCA and these related methods can be used as covariates in our machine learning approach, or simply regressed out of our feature data to begin with. Finally, a major challenge in genomic applications of machine learning is that we often don't have perfect data. And specifically, we often haven't measured the exact outcome that we want to learn to predict, at least not on a large scale. So it's very hard to get sort of perfect, good labeled data for many of the prediction tasks that we would like to do. So we have to get creative with the use of noisy labels and in understanding the differences between the data that we have and what you really want to predict in a real population. And I'll come back to that in a moment. So I'm going to end with an example from my lab that will highlight a few of these considerations that I've raised. So in this application, our motivation was personal genome interpretation. So every individual has something like 50,000 rare genetic variants in our genome. So these are genetic variants that are found at a minor allele frequency of less than 1% in the population. And current approaches are not very good at predicting which of these rare genetic variants, particularly those in non-coding regions of the genome, are likely to actually have a functional impact on gene expression or downstream on health. And this is a really important problem in the world. Currently, something like half of individuals with a rare genetic disease, when they go for clinical genetic interpretation, don't receive any genetic diagnosis. That is, we are unable to, with exome sequencing and standard approaches, identify the genetic variant that's causing their disease. So our goal here was to develop a machine learning predictor of rare variant function based on features from whole genome sequencing and RNA sequencing. So the approach here that we've taken is to build a tailored probabilistic model. The idea behind this is that if a rare genetic variant has an impact on someone's health, it's also likely to have a molecular signature in that affected person. So in addition to collecting their personal genome or their DNA sequence, we are also going to make use of personal functional genetic data, such as RNA sequencing, proteomics, or epigenetics, and build a prediction function. So this is a supervised task in the end, to build a prediction function that can take both DNA and RNA information and predict which variants are likely to be functional. So our outcome variable here is actually whether or not a genetic variant has functional consequences. So we designed a custom probabilistic graphical model that we called Watershed that is able to integrate personal genetic information along with functional data such as RNA sequencing. So in our probabilistic model, we capture both G, features from whole genome sequencing or annotations of the rare variant that we're trying to make a prediction for, along with E, or these are measurements from individual RNA sequencing, proteomics, epigenetics, or other functional data. And what we're trying to predict is actually an unobserved variable encoding whether or not this rare variant has an effect. So even though I cast the task as an unsupervised, I cast the task originally as a supervised problem, we'd like to predict whether or not a variant has a functional impact. In actuality, we don't have access to very good training data that indicates whether a genetic variant has a function or not. So we don't have really large-scale unbiased training data to actually perform a supervised learning machine learning task in this case. We simply haven't annotated enough rare genetic variants as functional or not functional, especially for the rare non-coding variants that we're most interested in. So we recast the problem as an unsupervised approach. So in this case, we don't require any labeled training data at all, indicating which variants are functional and which are not. So we cast this as a probabilistic model and learned it in an unsupervised scheme. It's a very efficient approach, so optimizing the model parameters using approximate inference takes just a couple of minutes. And from this, we're actually able to score any rare variant in a new patient based on any measurements that we have from that patient, whether it be their whole genome sequence alone or their whole genome sequence plus RNA-seq or plus RNA-seq and proteomic, et cetera. So what were some of the challenges that we faced as we designed this? I've already discussed the lack of training data available for casting it as a supervised problem, but we also did have to deal with some of the challenges I mentioned previously, such as confounders. So gene expression data, as I mentioned, has many technical and biological confounders, and these specifically obscure the effects of genetic variation. So genetic variants tend to have a small effect in their impact on gene expression compared to things like batch and age and even ancestry in some cases. So here we did apply methods including PCA or PEER to the gene expression data ahead of time as a pre-processing step before we input it as a feature in Watershed. And we did apply PCA to the genotype data to identify and account for genetic ancestry. And finally, we also threw out samples with broad evidence of technical artifacts. So some individuals had such different gene expression profiles from the average population, sort of genome-wide, that we suspected that they had major technical artifacts in them, and a small number of samples were simply thrown out so as not to bias the rest of the learning procedure. Another challenge is the limited number of samples that were available, especially when we first developed Watershed, that have both whole genome sequencing and functional data available. So here, we did apply some regularization or shrinkage methods for all the parameters learned in the Watershed model. We also used an approach that allowed us to share data across multiple different settings. So we learn a unified model across all genes and all individuals, and across, actually, in this case, multiple tissues and even modalities to increase sort of our effective sample size. And finally, we've been working on this for a number of years, and the sample size has simply naturally increased. And so data availability has really helped. And because I mentioned that there's not really a good catalog of which rare genetic variants actually impact gene expression, it's not only hard to train a supervised method, but it's also hard to evaluate it. So I mentioned that it's difficult to evaluate unsupervised approaches, and that did hold here. So we had to take a very creative approach where we actually took individuals who have matching genotype to each other. So we call this our N of 2 genotype, genetic variants. So we have genetic variants where, exactly 2 or just a handful of people share that same genetic variant. And what we actually do is make a prediction using Watershed for one of those individuals for that exact variant using their whole genome sequencing and that first individual's functional data, such as RNA sequencing. And we use the second individual's functional data, such as their RNA expression profile, to evaluate whether the Watershed prediction is actually evident in the effects on the expression level in that second held out individual. And we call this our N of 2 evaluation. But we really did have to get creative with how we evaluated this approach. But using that particular evaluation scheme, we did show that Watershed dramatically improves our ability to identify rare variants with functional impact once we include RNA sequencing over just whole genome sequencing alone. And that's evident in the signals for total expression, for allele-specific expression, and for splicing in the transcriptome. So Watershed does offer large improvements over whole genome sequencing alone. But in this case, especially given the evaluation that I described being a little complex and creative, we definitely needed to replicate this in independent data and actually go through with experimental validation and validate the predicted impact of several variants using a CRISPR experiment. So to show one final result, this did in fact replicate well across cohorts, and this is actually a result that Casey Brown helped us collect in a separate cohort. So using a Watershed model trained in GTEx, but applied in a separate cohort, we showed that the predictions made based on GTEx hold up nicely in the separate cohort that we evaluated, showing that individuals with a high Watershed posterior of having a large effect size in blue are much more likely to have strong effects evident in their RNA sequencing compared to individuals that had a low Watershed posterior. Okay, so with that, I would just like to thank everyone who contributed to the Watershed model from my lab, and I've highlighted some of the leaders of the development of that method. And I'd also like to thank my collaborators from the GTEx Consortium who worked on the Watershed project with us and also on the overall GTEx work that we've done over the recent years. And I specifically want to thank Casey Brown for his contribution to our work and really more broadly for sharing his scientific vision with me and with the genetics community. He's had a tremendous influence on how we think about genetics and the genetics of gene expression, and particularly in interpretation of GWAS and post-GWAS frameworks. So thank you very much to Casey.